So good morning all. I welcome you all to my channel Study Ka Enzyme, and today we are here to present you with the part 2 of Heredity and Evolution. So let's get started. Once we have done in the previous part, we have done part number 1, where we discussed about what is heredity, what is evolution, and we had also discussed certain terms like phenotype, genotype, heterozygous, homozygous. So now we will aware you about the most important part of heredity and evolution, which is monohybrid and dihybrid crosses, that I told you earlier also that they are very important and might come in your exam, in your board exam, your pre-board exam. So let's begin the chapter and begin with those crosses. So monohybrid as the name suggests, monohybrid cross means, monohybrid cross will mean, mono means one, and hybrid cross where we refer to only one gene being talked about. When we talk about only one gene, for example, tall and dwarf, when I take two pea plants and I cross one tall pea plant with another dwarf pea plant, so such a cross will be called as a monohybrid cross, as you can see on the screen as well. A tall plant, homozygous tall, crossed with a homozygous dwarf plant. Similarly, these crosses can also be between uh, two round and wrinkled plants. So first is the law of dominance and second is the law of segregation. As you can see, we are segregating the two genes. You know these two genes are present on the two homologous chromosomes that I am drawing on the screen. One chromosome is from the mother, another one is from the father. So one gene will be capital T present on one chromosome, the other will have other capital T, right? And for there, during the cell division, these chromosomes get separated into the gametes. So same ways we have law of segregation separating the two genes. Alright, so you also must have understood. Same ways we do it for small t. These two small t's, like the dwarf genes, also get separated from each other because of the law of segregation, because of the separation of gametes, formation of gametes. So now you can see on the screen I'm drawing a Punnett square where I put on the upper and the lower side, that is on the column and the row side, the two gametes, capital T, small t and small t. Now we fill this matrix. So when we are going to fill this matrix, so we will see it becomes capital T, small t for all the four squares. That means the F1 generation which is formed here. The initial one was the parent generation or the parental generation, capital T, small t. But the F1 generation which is formed here will all be heterozygous. As you can see, one capital T and one small t, which means they are heterozygous, and their phenotype will be tall. They will always be tall, 100% tall. So this question might also come in your exam. What is the genotype of the F1 generation progeny? So the answer is capital T, small t. What is the phenotype of F1 generation? So phenotype will always be tall. Alright, so this is the F1 generation. Now, when I self-cross these F1 progenies, that is, the generation obtained in the F1 cross, so capital T, small t, capital T, small t, when I cross these, self-cross these or self-pollinate these plants, so you will be able to see that these capital T and small t will again fertilize, and now we will have separation of gametes as we saw earlier. Let me add a slide here, a blank slide. Okay, now you will be able to see that this capital T and small t will get separated into capital T and small t. And in the same way, we will fill the Punnett square. So we will be filling up the Punnett square just like we did in the previous case. And now you see the F2 progeny, the result which is formed now will be the F2 progeny. Okay, so this F2 progeny will show a different result as compared to the F1. I'm making a Punnett square here, and now if you see, the result will be capital T small t, capital T small t. So when you fill up these, it becomes capital T, 2 capital T and small t, and single small t small t. So this small t is homozygous dwarf. Capital T will be homozygous tall. Very good. And we have two heterozygotes. That is two heterozygous conditions, capital T and small t. So the small t will lead to a dwarf formation, dwarf individual. Capital T small t, because of the law of dominance, capital T will mask over the other one. So all these three, capital T, capital T small t, all these three will now become tall. So if I ask you to write the genotypic ratio, so the genotypic ratio will become capital T, capital T small t, because we have these three type of combinations only, and small t. So we have one capital T, right? Two capital T small t, and a single small t. Alright, so the genotypic ratio will always be 1:2:1 in case of the F2 generation. Let me remind you, in the F1 progeny, it was all capital T and small t. But when we self-cross these, now it became capital T, capital T, capital T and small t and small t. Alright, so if I talk about the phenotypic ratio, now you can see 3 tall and 1 dwarf. So the phenotypic ratio that you see, the phenotypic ratio will be tall is to dwarf will be 3:1. Okay, so it will become 3:1. Okay, 3:1 we have to remember this. Had it been a yellow seed and green seed, so isme yellow seed is dominant, so we would have got 3 yellow seeds and 1 green seed. Okay, same ways, had this cross between purple flower and a white flower, this would have become 3 purple and 1 white. So recessive 1 will be 1 and capitals will be, dominant will be 3 in the phenotypic ratio. Alright, so we have seen two laws here as of now. One is law of dominance, which states that the capital gene or the dominant gene will express itself over the other one. Okay, and then is the law of segregation, right? Law of segregation states that the capital T and small t will get separated. So here you can see me highlighting those T, capital T, all the tall generations, the phenotypic ratio. Okay, same way there's another cross between capital T small t and small t. So we see separation of gametes. So gametes get separated. This is law of segregation, and you can fill up this Punnett square. All right, then we have dihybrid cross. Now when I talk about a dihybrid cross, di means two, hybrid when I talk of two genes. So when I cross two individuals and consider their two genes, for example, tall and round seed. Okay, so this will become a dihybrid cross. Here in the example which is shown here, we can also show the cross between any two genes. For example, round seeds, ringle seeds, yellow seeds and green seeds. We can also do one example between tall and dwarf, yellow seed and green seed. We can also do one cross between tall and dwarf. So this is one gene, and the other one will become, yes, round and wrinkled. Anything. I had told you we have 7 contrasting traits. So these you can pick any two genes and any two alleles. Any two alleles. And for the two genes it becomes 4 alleles. As you can see on my screen, I'm drawing a dihybrid cross between a tall and a round plant, and the other plant is dwarf and wrinkled. Okay, so the first step, as we did in the monohybrid cross also. Let's do that. We will separate these two into two gametes. So T and R. We will not separate TT and RR. Please don't do this mistake. Be very mindful. We will be separating the two genes together. So it becomes capital T, small tr, small tr. Okay, so this is the parental generation. Now when we talk of the first filial generation, so again we will make a Punnett square, and we will write these genes or the gametes that we that have formed on the row and the column side to fill up the matrix. So on the left hand side we will write capital T R, capital T R. On the upper side we will write TR and TR. Okay, so now when I fill up, it becomes capital T, small t, capital R, small r. Same here for all the four uh, boxes that you see on my screen. So it is again 100%. F1 generation is 100%, as we did for the monohybrid also. Same ways for the dihybrid cross also. We have both, right? F1 generation having 100% dominant appearance. Okay, in the monohybrid it was tall, and here we have both the tall genes appear or dominant genes appearing. For example, tall and round. So all the four 100% capital T, small t. So it will appear, you tell me it's a heterozygous thing, it will appear tall, and capital R and small r, it will become round. So now we have F2 generation. We will be making up the F2 generation from the F1 generation on self-pollinating or selfing F1 generation. I've written selfing, but it actually means self-pollination when a plant, when the anther of the same anther and the pollens from the same plant are sprinkled over the stigma of the same. So we have self-pollination. So genes will remain the same, combinations will remain the same, capital TR. पर यहाँ अगर आप देखो, so we have four new combinations forming. Capital T, capital R, capital T, small t, capital R, small t, small r. Again the same thing here. ठीक है? The combinations will all be same. Capital T, small r, capital T, capital R, capital small t, capital R, small t, small r. So again we will make a big Punnett square now, which is 4 into 4, that is 16 square boxes वाला एक Punnett square बनाएंगे. I think I'll make it on the next page. This page is too small to do that. Okay, so if I do it on the next page, so we will make a 4 into 4 matrix, or column side में I will write the gametes. Okay, and on the row side also I will write the four combination of gametes. Okay, and I want you all also to pause this video and just do it on your own. ये आप एक बार box बनाओ. This is extremely important, guys. If you do it, you will understand it, because the same concept is there in your 12th also. Okay, so capital T, capital R, capital T, small t, capital R, small t, small r. और ऊपर column वाली side में भी we will write the same combinations. Okay, so we are going to write the same combination, that is capital T, capital R, capital T, small t, capital R, small t and small r. Okay, so now we will start filling up the matrix. So I want you all to do along with me. Pause this video and do it, and see who does it first. आप पहले करते हो कि मैं? Let's see that. Okay, so I am filling up this capital T, capital R, capital T, small r. So make sure that you write both the alleles together. जैसे आपको पता है ना, tall की gene, tall, dwarf alleles हैं, राउंड रिंकल्ड एलील्स हैं. सो ट्राई टू राइट इट टुगेदर. इससे आपकी कंफ्यूजन कम होगी और एरर भी कम होगा. ठीक है, सो कैपिटल टी, स्मॉल आर, कैपिटल आर, स्मॉल आर. सो प्लीज फिल अप द एंटायर matrix as I'm doing here, and please do along. As much as you practice, the more efficient you become, right? Forgive me for my scribbling. I'm new to this pen tab. I'm sorry. Okay, so this is happening, and then we will be separating these phenotypes, because genotype separate करना थोड़ा difficult हो जाता है. It will become capital T, small t, capital R, small r. Capital T, small t, capital R, small r. Capital T, small t, capital R, capital R. Capital T, small t, capital R, small r. You know how to fill up these matrix. So what we do, we will multiply these. ये जहाँ पे हम, जैसे आपने देखा होगा लैटिट्यूड और लॉन्गिट्यूड्स जैसे ग्लोब में क्रॉस करते हैं, देन वी लोकेट वन पोजीशन ऑन द ग्लोब. सेम वेज अगर ये रो और कॉलम, इनको हमने मल्टीप्लाई करना है. दैट इज मल्टीप्लाइंग, मतलब हम इनको ऐड कर देंगे. एक ऊपर का टी, एक स्मॉल नीचे का टी. एक फॉर एग्जाम्पल the last one. So we have small t on the column side, small t on the row side. So it becomes small t, small r on the row side, small r on the column side. So it becomes capital T, small r, small r. Now I want you all to tell me the phenotypes. As you can see, I'm highlighting with yellow color the ones who have the phenotype tall and round. चाहे वो कैपिटल आर हो वो भी राउंड होगा, कैपिटल आर स्मॉल आर हो वो भी राउंड होगा, कैपिटल टी वो भी टॉल होगा, कैपिटल टी स्मॉल टी वो भी टॉल होगा. सो आई एम एनसर्कलिंग द वंस विच आर एक्चुअली, ये गलती से हो गया, आई एम सॉरी, इट शुड बी स्मॉल आर. ओके, आई विल जस्ट रेक्टिफाई दैट. और इसमें हम सिर्फ उन्हीं को येलो से सर्कल करेंगे सो दैट काउंटिंग बिकम्स इजियर फॉर अस. काउंटिंग बिकम्स easier for us. Okay, and now you've seen जितने भी कैपिटल टी स्मॉल आर हैं, कैपिटल मतलब जिनका फिनोटाइप टॉल और राउंड है, मैंने उन्हें येलो कलर से सर्कल कर दिया है. नाउ आई विल यूज अ डिफरेंट कलर फॉर एनसर्कलिंग tall and wrinkled. Okay, so I will use a different color palette. I'll use green color. T, capital T and small r. So it becomes tall and wrinkled. Tall and wrinkled. So we have three combinations becoming tall and wrinkled. Becoming tall and wrinkled. Now I will use a different color to highlight dwarf and red. Dwarf and round. Dwarf and round. So we have three combinations having dwarf and round combinations, and I use the last sky blue color to highlight dwarf and wrinkled gene. Okay, so now if you look at the four combinations, you will be able to see कि tall और round है 9, tall और wrinkled है 3, dwarf और round है 3 और tall और wrinkled है 1. Now just tell me what was the initial combination. So initial combination was tall and round on one side and dwarf and wrinkled on the other side. But what are the combination that you get in the F2 progeny? अगर मैं F2 progeny की बात कर रही हूँ, यानी कि F2 generation की बात कर रही हूँ, so I am able to see 9 tall and round, which was there one of the parental combination. I get tall and wrinkled 3, which is a new combination. Dwarf and round 3. इसका मतलब कि ये दो नए जीन्स हैं क्रॉसिंग के ऊपर, रिकॉम्बिनेशन के ऊपर. जैसे क्रोमोसोम्स क्रॉस ओवर करते हैं, आपने पढ़ा होगा कि जब सेल डिवीजन होता है, मियोसिस होता है, तो उसके अंदर क्रोमोसोम्स पैकेटिंग स्टेज में क्रॉस करते हैं और उसकी वजह से नए जीन कॉम्बिनेशंस बन जाते हैं. और ये नए जीन कॉम्बिनेशंस जो बने हैं, दे आर टॉल एंड रिंकल्ड. इसका मतलब टॉल और राउंड की जीन्स पहले एक क्रोमोसोम पे साथ में थी, ठीक है. ड्वार्फ और रिंकल्ड की जीन एक साथ थी, बट एज यू गेट टू द F2 जनरेशन, ये जीन्स आपस में सेपरेट हो गई हैं. इसका मतलब जो जीन इनिशियली साथ में थी, जरूरी नहीं वो एंड तक F2 जनरेशन तक साथ में रहे. ओके, दिस इज लॉ ऑफ सेग्रीगेशन. नाउ लेट्स लुक एट अनदर एग्जांपल, round and yellow. So round is, you know, is a dominant trait and yellow is also a dominant trait. The other one, green matter, wrinkled वाले, so that's a recessive trait. अब आप कहोगे कि मटर तो green ही होते हैं, but we talk of these mature pea seeds, okay, dried and mature, तो ये yellow हो जाते हैं. Alright, so same, I want you all to pause this video and do this again. Now you have done one example with me. Now I want you all to do this with me. चाहे कोई भी आप क्रॉस करा लो, हमेशा जो आपका कॉम्बिनेशन होगा वो 9:3:3:1 ही आएगा इन केस ऑफ अ डाइहाइब्रिड क्रॉस F2 का रेशो. बट इन केस ऑफ अ मोनोहाइब्रिड क्रॉस, यू नो F2 का रेशो क्या होता है, 1:2:1. जीनोटिपिक और फिनोटिपिक का रेशो होता है 3:1. ओके, तो यहाँ पर भी 9 वाला होगा पेरेंटल, डोमिनेंट वाला पेरेंटल, 3 वाला न्यू कॉम्बिनेशन, 3 वाला न्यू कॉम्बिनेशन, और जो लास्ट में 1 आता है वो होगा रेसेसिव वाला पेरेंटल कॉम्बिनेशन, रेसेसिव ट्रेट. चाहे पेरेंटल कुछ भी हो, बट यू ऑल नीड टू अंडरस्टैंड, अगर दो जीन्स के बीच में क्रॉस हो रहा है तो maximum 9 number will be the dominant genes, both the dominant genes. 1 will be both the recessive genes, and 3 will be the new combinations or the heterozygotes, जहां पर एक dominant होगा, एक recessive होगा. All right. अब ये डोमिनेंस और रेसेसिव जीन होती क्यों है, जस्ट अंडरस्टैंड दैट. तो वो अंडरस्टैंड करेंगे हम कैसे, जैसे आप देखते हैं एक फैमिली में ब्लू आई कलर है और एक ब्राउन आई कलर है. इफ यू लुक एट दीज, यू नो आउट ऑफ दीज ब्राउन आई विल बी मोर डोमिनेंट. इसको हम डोमिनेंट कैपिटल बी से डिनोट करते हैं और ब्लू आई को हम स्मॉल बी से डिनोट करते हैं. बट डू यू नो व्हाई, what is the difference between this dominant and a recessive trait? I'll tell you. आपको पता है कि जीन्स, डीएनए का काम होता है आरएनए बनाना, आरएनए का काम होता है प्रोटीन बनाना. तो जो भी आई कलर का प्रोटीन होगा, ठीक है, वो अगर कंप्लीट बना रही है एक जीन, फुली फंक्शनल जीन, तो वो क्या बनेगा, डोमिनेंट. और अगर वो इनकम्प्लीट बन गया, अगर वो जीन रेसेसिव है, तो वो प्रोटीन कैसा बनेगा, इनकम्प्लीट बनेगा. ओके, नाउ दैट यू सी, चाहे वो अगर अनमॉडिफाइड जीन होगी तो उसका प्रोटीन कंप्लीट होगा, ठीक है, और वो जीन कैसी होगी, डोमिनेंट. पर अगर मॉडिफाइड जीन हो, किसी भी कारण से मॉडिफाइड जीन, तो उसका प्रोटीन या तो ट्रंकेटेड बनेगा, मतलब छोटा बनेगा, या फिर इनएफिशिएंट इन फंक्शन बनेगा, बिकॉज ऑफ विच द रेसेसिव जीन विल नेवर बी एबल टू एक्सप्रेस इटसेल्फ. पर एक हेटेरोजाइगोट की अगर बात करोगे तो दोनों होंगे उसमें, डोमिनेंट एंड रेसेसिव. तो एक भी अगर प्रोटीन नॉर्मल बन रहा है, सो फिनोटाइप विल बी नॉर्मल. दैट इज वाई हेटेरोजाइगोट का phenotypic appearance dominant वाला होता है, ठीक है. Then you have law of segregation. Now by this time you also must have understood that we have two homologous chromosomes in all our cells, one from mother, one from father. So these chromosomes, they get separated. As a result, the genes which are present on them also get separated. When these genes get separated, we call this condition to be law of segregation. Now if I talk about the third law, लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. इंडिपेंडेंट मतलब इंडिपेंडेंट ऑफ द अदर. सो अगर हमने यहाँ पे डाइहाइब्रिड के केस में देखा था कि दो पेरेंटल कॉम्बिनेशंस अगर साथ में थे, ठीक है, अगर दो पेरेंटल कॉम्बिनेशंस, लेट मी हैव अ लुक एट दैट अगेन. तो अगर दो पेरेंटल कॉम्बिनेशन साथ में थे, ठीक है, अगर वो साथ में है तो इनिशियली वो होंगे पेरेंटल जेनरेशंस, और दो नए कॉम्बिनेशंस जो बनेंगे वो होंगे न्यू कॉम्बिनेशंस. दोस पेरेंटल कॉम्बिनेशंस एंड न्यू कॉम्बिनेशंस टुगेदर विल फॉर्म व्हाट, लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. टुगेदर विल फॉर्म लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट का क्या मतलब हुआ कि जो जीन शुरू में साथ में थी, पेरेंटल कॉम्बिनेशन में साथ में थी, जरूरी नहीं है कि F2 जनरेशन तक आते आते दे गो टुगेदर, they can get separated. यहाँ आपको स्क्रीन पे दिख रहा है कि कैपिटल A एंड स्मॉल a है एक क्रोमोसोम के ऊपर, और दूसरे क्रोमोसोम पे कैपिटल B स्मॉल b, ठीक है. अब जब आप इसे देखोगे सेपरेशन के लिए, तो ऐसा भी हो सकता है कि कैपिटल A के नीचे स्मॉल b आ जाए, कैपिटल A के साथ स्मॉल b रहे. So there are two new combinations which are formed. All right. So this is because of the recombination, right? So I hope you would have understood this law of segregation. लॉ ऑफ सेग्रीगेशन मतलब जो दो जीन्स नॉट एलील्स जो इनिशियली साथ थे जरूरी है लास्ट में साथ रहे. देन यू हैव सेक्स डिटर्मिनेशन इन केस ऑफ ह्यूमंस. we'll discuss about it little later in detail. अच्छा, now if I give you one example, dihybrid क्रॉस को आपको अगर करने को दिया जाए एक टॉल और राउंड, उसका क्रॉस करें आप ड्वार्फ और रिंकल्ड से, can you do it with me? So will you write it? What will you write the genotype? It will be tall. टॉल को हम कैसे लिखेंगे? टॉल राउंड. अगर ऐसा होगा तो इसकी F2 प्रोजनी क्या होगी? टॉल राउंड 9, टॉल रिंकल्ड वो क्या हो जाएंगे? 3, क्योंकि ये न्यू कॉम्बिनेशन है, वन फ्रॉम वन पेरेंट अदर वन फ्रॉम अदर वन. ड्वार्फ और राउंड क्या हो जाएंगे? 3. और जो होमोजाइगस रेसेसिव है, दोनों के रेसेसिव ड्वार्फ एंड रिंकल्ड, इट विल बिकम 1. ओके, तो ये कॉम्बिनेशन हमेशा सेम रहने वाला है. 9:3:3:1 आप हमेशा के लिए याद रख लें. ठीक है, चाहे कोई भी जीन हो, कौन सी भी जीन में क्रॉस हो रहा हो, जो डोमिनेंट वाली होगी इट विल बी 9. डोमिनेंट होमोजाइगस 9, डोमिनेंट सॉरी रेसेसिव होमोजाइगस 1 और बीच का हेटेरोजाइगोट्स होंगे, they will be 3 and 3. I'm sorry, please forgive me. It's a little late at night, so I'm recording this video. Okay, all right, now let's move ahead, and I hope by this time you all would have understood all three generations, all two generations, and all the three laws where I'm showing you here two parental and new combinations. तो पहला लॉ बताएंगे आप? Yes, law of dominance. दूसरा law of segregation. Wonderful. तीसरा law of independent assortment. Now, if I talk about law of segregation, मैं आपको समझा ही चुकी हूँ. ठीक है, अब मुझे एक बात बताइए, which parent are you more like? Can you mention that in your comment? Or you can also give me some comment like you have your eyes like your mother, your hair color like your father. ठीक है, so just write that in comment. I'm waiting. Now we have acquired versus inherited traits. Acquired traits क्या होते हैं? That you gain in your lifetime, that you learn, that you build in your lifetime. Inherited मतलब that you have received from your parents and they are with you since the birth. जैसे आपका eye color, आपका hair color, वो आपके पास traits हैं since the birth. ठीक है, you have acquired these. Shape of your fingers, shape of your nails. So these are some of the traits that you have acquired since your birth from the parental generation. और acquired traits होते हैं जो आप सीखते हैं अपने लाइफ स्पैन में. जैसे मैंने बैडमिंटन सीखा, ठीक है, मैंने अच्छी हैंडराइटिंग सीखी, I learned music. Okay, so you understood this traits acquired and inherited. See what I have for you. I've made a family tree. Let me get it back to you. I have made a family tree of my family where I represented my grandparents, my parental generations, my papa, chacha, and my papa, chacha, Sanjay chacha, Goli chacha, and their families. So same ways you can make your own combinations also. You can make your own family tree also. It will be very nice doing that. Okay, now one thing that we are left with is sex determination in humans. So you have only sex determination in humans. वो मैं अभी इस ट्री के बाद डिस्कस करती हूँ. My grandfather, my grandmother, they had 4 children. ताऊजी did not marry, so आगे उनका family tree नहीं बनाया. Then 5 father, my mother, and we 3 siblings. Sanjay Chacha, Chachi, 2 siblings, Sunny and Kaku. Me, my sister Neha, and my brother Gaurav. Goli Chacha and Chachi. Then I have 2 cousins, Shubhi and Shubham. So isn't it interesting to know the family, making a family tree? और humans की अगर हम बात करें, so you know we have two sex chromosomes, XX and XY. So XX chromosome is in the female and XY chromosome is in the males, as you can see here. So XX is in female and XY is in male. So these also get separated, जैसे हमने law of segregation किया था. So XX will get separated, X and Y will get separated. तो इनका भी जब हम Punnett square करेंगे, so we will be able to see we get 50% XX and 50% XY. XX मतलब it will be a female, XY it will be a male. So there are equal chances, that is 50% chance of the gender of the child being a boy, 50% chances that the gender of that child will be a female. I mean, it will be a girl child. So there are equal chances. So there used to be a value-based question some years back also, that a family is cursing the women for giving birth to all the female child. तो उसका आंसर क्या था कि who is responsible for that? So Y chromosome is responsible for sex determination in case of humans. Okay? और क्योंकि Y chromosome is from the father, so he decides the gender of the child, not the female. Okay? और एक और इंटरेस्टिंग बात बताऊं, देयर आर सम एनिमल्स जिनमें एनवायरमेंटल फैक्टर, टेम्परेचर डिसाइड करता है कि गर्ल फीमेल प्रोजेनी होगी कि मेल. Okay, now I want you all to go and search about how is the gender of lizards and crocodiles decided. Okay, please write to me in comment in case you see that. It's very interesting, you will find them interesting. क्रॉस करके बताया था XX, XY. अब हम आ जाते हैं mnemonics के ऊपर. Tricks and mnemonics to learn this chapter. Okay, let me put it ahead. ये कुछ questions हैं. Please go through these questions, pause this video and look at these previous year questions that I have written for you. और ये कुछ questions हैं जो आपको prepare करने ही चाहिए. Variations क्या होते हैं? Variations are the differences that you see. अच्छा, मेंडल ने खाली इसको ही क्यों चूज किया, पी प्लांट को? Contrasting traits की वजह से. You can see on the screen, these are some of the mnemonics. LAD means law of dominance, law of independent assortment, and D, law of segregation. Segregation मतलब divide. तो LAD याद रखेंगे, so you will be able to remember these things. Okay? LAD always wins. तो इसका मतलब dominant will win over the recessive ones. ठीक है, डोमिनेंट को हम कैपिटल लेटर से डिनोट करते हैं और रेसेसिव जीन्स को हम स्मॉल लेटर से डिनोट करते हैं, है ना? Okay, another trick. Big personalities dominate the room. तो इसका मतलब big personalities यानी कि big alphabets will dominate. तो big characteristics will be dominating. ऐसे genotype versus phenotype. G के अंदर ही no आ रहा है और pheno उसके अंदर yes. तो मतलब क्या? Genes you got that from your parents, तो वो no मतलब they are not visible from outside. So what you can see in an individual is not genotype, rather जो उनकी physical appearance है जो हमें देखने को मिलती है वो क्या होती है? उनकी phenotype. Okay, so this was all about it. There were some questions that I told you earlier. Please subscribe to my channel and like it, and I am working very hard every day and improving myself. Please support. Thank you.